Hello everyone, today we're going to talk about microbial genetics. The genetic information stored within the DNA is what's going to determine which proteins are going to be made. So when we think of the central dogma, you think of how all the information is stored within the DNA. So basically the DNA acts as the blueprint. This DNA is going to be transcribed into a messenger RNA. So that process is going to be the transcription process. And then that messenger RNA is going to be translated into the proteins. So whenever a protein is made, transcription, then translation will need to take place within the cell. Bacteria do have their own DNA. Their DNA is usually circular chromosome. Bacteria, they could also have plasmids, and plasmid is another set of DNA, circular, and plasmids, they could carry genes that could code for antibiotic resistance. Let's take a look at the structure of the DNA together. So DNA is double-stranded, so you can see both strands of the DNA right here. The backbone of the DNA is made up of the sugar group and the phosphate group. So you can see right here, both backbones, they have the sugar groups attached to the phosphate groups. And then right here, attached to the sugar groups, you have the nucleotide bases. And the bases for the DNA are thymine, adenine, guanine, and cytosine. And you can see that those bases are linked together. Adenine always binds with thymine, and guanine always binds with cytosine. The DNA has two ends. One is called 3' and the other end is called 5'. So each DNA strand has both the 5' end and the 3' end. The 3' end is usually the end that has the hydroxyl group, the OH group attached to it, and the 5' end is where you have the phosphate group. Both DNA strands are anti-parallel. And the reason why we call them anti-parallel, because you can see that on this side you have the 3' end, and on that side it's flipped. So you have right here the 5, and then up here is the 3' end. And so that's why we say that our DNA strands are anti-parallel, and also the DNA strands are complementary to one another. They're not identical. So this strand right here is complementary to that one. It is not identical. Bacteria cells, before they multiply themselves, before they grow, what they have to do is duplicate their own DNA so that when they split, they're going to have their DNA. That process of duplicating their DNA is known as DNA replication. If we zoom in on the DNA right here of the bacteria, you can see that it's a circular DNA. It's double-stranded, so it's made up of two strands. Right here within the DNA, there is a region known as the origin of replication, and this is where the replication process begins. You can see that both strands are going to start to unravel, and both of them are going to start to replicate. So you are having bidirectional replication taking place, and both strands are going to start replicating until we hit this region right here, the termination of the replication, and both strands are going to detach from one another. And what you're going to end up with is one old strand that's now has been replicated, and you have the new complementary strand. And of course, same thing here, one old strand and its new complementary strand. Which enzyme helps build that new complementary strand, by the way, and it helps bring and build those nucleotides, an enzyme called DNA polymerase. And the replication process, it's highly accurate, So very minimal, almost very, very low percentage of error takes place during DNA replication. And that's because the DNA polymerase is really good, and it has high proofreading capability. So when that DNA polymerase is building the new strand, it's making sure that adenine is going to bind with thymine and guanine is going to bind with cytosine, and it proofreads it, so it makes sure that everything is okay. That's why, again, DNA replication is a very highly accurate process. When DNA want to make their own proteins, they will have to go through the transcription process, and then they're going to have to go through the translation process. So let's go ahead and begin with the transcription process that's going to take place within bacterial cells. Transcription is the process of transcribing the DNA into messenger RNA. Now, transcription begins when an enzyme known as RNA polymerase binds to a specific region within the DNA sequence known as the promoter sequence. Once the RNA polymerase binds to that promoter sequence, then transcription can go ahead and proceed. If the RNA polymerase is not able to bind to that promoter sequence, then transcription is going to stop. And this is actually one way for transcription to be regulated within the prokaryotes, is to go ahead and prevent the RNA polymerase from binding to the promoter sequence and therefore stopping transcription. And we're going to talk about a few examples. But if RNA polymerase is not prohibited from binding to the promoter sequence, then transcription does take place, and transcription is going to start, as I said, building that messenger RNA sequence. The messenger RNA sequence, by the way, is single-stranded, so it's not double-stranded like the DNA. And when the messenger RNA is built, it's always going to proceed in the 5' to the 3' direction. And only one DNA strand gets transcribed. So as we said, DNA is double-stranded, so you have 2 strands. When we talk about DNA replication, both DNA strands get replicated. But when we talk about transcription, only one strand of the DNA is going to be transcribed and result in the production of the messenger RNA. Now, transcription starts, as I said, within a sequence on the DNA known as the promoter, and it's going to stop when we hit a region within the DNA sequence known as the terminator sequence. So if we take a look right here, the DNA strand, the DNA, it's double-stranded, so you can see both strands of the DNA right there. As I said, transcription is going to begin when the RNA polymerase, represented by that bubble that you are seeing right here. So this is representing the RNA polymerase, which is the enzyme. Transcription begins when the RNA polymerase binds to a region within the DNA sequence known as the promoter. Once RNA polymerase binds to the promoter sequence on the DNA, transcription goes ahead and begins. And as you can see, the DNA is double-stranded. Only one strand is going to be transcribed. And the RNA polymerase is going to glide along the DNA sequence. And as the RNA polymerase is gliding along the DNA sequence, the messenger RNA is going to start getting built. And so if the DNA strand reads as you can see right here, thymine, then which nucleotide is going to come and bind to thymine? Adenine. If the DNA strand reads adenine, uracil is going to come and bind to adenine. The thing about RNA is that RNA is single-stranded, and RNA doesn't carry thymine. So instead of thymine, it carries the base known as uracil. So anytime the DNA reads adenine, a uracil RNA nucleotide base is going to come and bind to it. And as you can see, as the RNA polymerase glides along the DNA strand and the messenger RNA keeps getting longer, this transcription process is going to continue until a region on the DNA sequence known as the terminator sequence is reached. Once we reach the terminator sequence, then transcription comes to a stop. RNA polymerase just moves away, and the messenger RNA goes ahead and gets released. And during that process of transcription, as you can see right here, to transcribe the DNA, that double-stranded unravels itself, but as the RNA polymerase keeps moving along the DNA just goes ahead and winds back. As if, like, nothing happened, it can go ahead and wind back. And then when the transcription process has ended, everything is going to look back as normal. So you will have the double-stranded DNA, and then the RNA polymerase is going to take off. That RNA polymerase can be reused for another transcription process. And then this messenger RNA right here, now it's going to be translated to provide us with the protein. The translation process, as I said, this is when the messenger RNA that was just made from the transcription process is going to be translated into proteins. Now, this messenger RNA we call it messenger because it is carrying the message, and this message needs to be decoded so that we can understand which protein is it coding for. And so the way to read a messenger RNA is you read it 3 letters at a time, which we refer to as codons. Each codon is going to code for a specific amino acid. Why amino acids? Because proteins are made out of amino acids. So during translation, you're basically trying to build the chain of the amino acids needed to build the protein. So as I said, the messenger RNA is read 3 letters at a time, and those 3 letters are known, or 3 bases, they are known as decodons. So if your messenger RNA reads U, then the amino acid that it codes for is phenylalanine. For example, if your messenger RNA reads C, then the amino acid that it codes for is proline. If it reads AUG, then this is the amino acid known as methionine. The translation process begins when you see this AUG sequence within the messenger RNA. And then translation is going to stop when we hit any of these stop codons right here on the messenger RNA. Let's take a look at the process of translation together. So after transcription has taken place and the messenger RNA is right here, this messenger RNA is going to move within the cytoplasm of the cell until it reaches the ribosome. So this is the ribosome right here. And you can see that the ribosome is made up of two subunits. And then these two subunits of the ribosomes, they're going to enclose that messenger RNA. And that messenger RNA, as I was saying, it's basically carrying the coded message that we need to translate to figure out which protein is going to be made. And so this messenger RNA is basically carrying the message in terms of codons. So as I said, the messenger RNA is going to be read 3 bases at a time. And for you to figure out which amino acid each codon codes for, you need to look at the chart that we just discussed. Now, there are actually 3 types of RNA. We have rRNA, and this is basically the RNA that you're going to find within the ribosomes. There is messenger RNA, and so here is the messenger RNA carrying the message. And then there's another form of RNA known as tRNA, and the T stands for transfer. And the reason why it's called the transfer RNA, because this RNA is going to be responsible in bringing over the amino acids. So as I was saying, when you look at your messenger RNA, the way you read it is you read the 3 bases at a time known as the codons. Then you refer to the chart to figure out which amino acid it codes for. So each codon codes for an amino acid. Now, which RNA is going to come and bring over that amino acid that's needed? That's the job of the transfer RNA. So you can see right here the transfer RNA, it's carrying the amino acid that's needed. And you can see that the ribosome is going to move along that messenger RNA, and multiple tRNAs, as you can see, multiple tRNAs, they keep coming over, and they go ahead and they bring over the amino acid needed, and then they take off. So this process keeps taking place. tRNAs, they keep coming over, bringing over the amino acid, and then they take off. And you can see that the chain of the amino acid keeps growing longer and longer and longer until we hit that stop codon on the messenger RNA. Once a stop codon is reached, then the process of translation stops. And you can see that the messenger RNA basically takes off. The ribosome also takes off, and it gets basically everything disassembles. And here is the final chain of amino acids, which is going to become the protein. This messenger RNA, can it be translated one more time? Yes, it can. This messenger RNA can go ahead and get translated one more time, and a new protein would be achieved. Same protein though. So this is the process of translation. Now, when we think of eukaryotic cells, when eukaryotic cells want to make their own proteins, they still have to go through transcription and translation, but transcription and translation in eukaryotic cells happen in order. So in eukaryotic cells, transcription must take place first, and it must be completed before translation begins. But that's a different story in bacterial cells. In bacteria, as the DNA is being transcribed and the messenger RNA is being formed, translation can actually go ahead and begin, even though transcription has not ended yet. So in bacteria, translation can actually go ahead and begin and proceed before you have the final messenger RNA sequence. Bacteria can actually control their transcription process. They can either repress it or they can induce it. If they repress transcription, that means that certain genetic sequences are going to be turned off and they are not going to be expressed. That's possible due to some proteins that are known as repressor proteins, and those repressor proteins can actually block transcription. Usually the default position for those genes is on, so usually those genetic sequences are being expressed and they are only turned off when a repressor is located. If the repressor is not there, then the genes are going to be transcribed. Induction, on the other hand, is turning on certain genetic sequences that are usually turned off. So again, this is initiated usually by an inducer, and that inducer goes ahead and switches the default position of those genes, which are usually off. They're usually turned off. That inducer goes ahead and turns on those genetic sequences to be translated into the proteins. Bacteria, such as E. coli, have what's known as an operon. What's an operon? An operon is basically a set or a group of genes that are usually transcribed together. So you usually have a bunch of genes, 3, 4, 5. They're usually a group of genes that are next to one another. If you look at the DNA sequence, they're usually grouped together. And those group of genes, they're usually transcribed together and therefore translated together. And those genes are controlled by a sequence on the DNA known as the promoter sequence. And we're going to talk about how do some of these operons work. There's also a segment within the DNA known as the operator, and the operator is going to be pretty important because this operator, if it gets blocked, if this sequence on the DNA gets blocked, then transcription stops. So let's go ahead and take a look at an example of an operon. As I was saying, an operon is basically a group of genes that are grouped together on a DNA sequence, and those group of genes, they're usually transcribed and translated together. And if we take a look at an operon, as I was saying, it's basically on the DNA sequence. So here's the DNA sequence. It's double-stranded. There's usually a region within the DNA sequence that's known as the regulatory gene. And then comes the promoter. Then comes a region known as the operator. And then comes the structural genes. And these genes, they are usually transcribed and translated together. The operator is where the repressor is going to bind. So if there is a repressor, then this is where the repressor binds. It binds right here to the operator. The promoter is where the RNA polymerase binds to begin transcription. So if that promoter is blocked, then transcription is going to come to a stop. And so that regulatory gene is what's going to determine if transcription is going to proceed or if transcription is going to be inhibited, because that regulatory gene can code and translate that DNA sequence into repressor proteins. There are two types of operons, inducible operons or repressible operons. Inducible operons, those are a group of genes that are usually turned off, so those genes are not being expressed, and they're only going to be expressed only if there is an inducer around. Once that inducer is around, then that inducer binds to the repressor protein that's blocking transcription. And once it binds to it, then the repressor is going to come off and transcription can go ahead and proceed. Irrepressible operon, on the other hand, that's an operon, which is, as I was saying, an operon is a group of genes. So they are a group of genes that are usually turned on and they are usually being expressed. And those genes are going to be repressed and turned off when a repressor is around or when a co-repressor is around. If these repressors are around, then those repressors are eventually going to bind to the operator sequence on the DNA, turning off and stopping transcription from taking place. And if transcription is stopped, then obviously translation is also going to be stopped. Let's take a look at an example of an operon. As I was saying, one more time, an operon is a group of genes that are usually transcribed and translated together. So E. coli has an operon known as the Lac operon. Lac is short for lactose. So E. coli has the ability to control the genetic expression of certain genes that are going to code for proteins that are going to metabolize lactose. These genes are either turned off or they are turned on. An inducible operon is usually a group of genes that are usually turned off. That's like their default. They're usually off, and they're going to be turned on and expressed if there is an inducer around. So let's take a look at the Lac operon together. So here is the double-stranded DNA. Here is the regulatory protein. This regulatory protein, when it's transcribed and translated, so you can see right here, it's being transcribed and you get the messenger RNA, and then it's translated. What is this regulatory protein going to provide us with or provide the E. coli with? It provides it with a repressor protein. So right here, the transcription and translation has resulted in the production of a repressor protein. This repressor protein is active, and what it does is it goes ahead and it binds to this sequence right here. This sequence is the operator sequence. So here is the regulatory gene sequence. Here is the promoter sequence. Here is the operator. And then here are the genes that are going to, in this case, help metabolize lactose. We give them letters. So this is gene Z, Y, and A. And those genes right here, as you can see, they are grouped together. Those genes are, if they are expressed, then they are going to help the E. coli. They're going to help the E. coli absorb the lactose and metabolize it and break it down. That's if they are turned on. But as you can see right here, they are actually turned off, and transcription of these genes and translation of those genes are inhibited by the repressor protein. The repressor protein usually binds to the operator sequence. Now, why is transcription and translation of these genes turned off? Because for transcription to begin, RNA polymerase will need to bind to the promoter sequence. And then it has to start to glide along the DNA sequence to go ahead and start the transcription. Now, do you see a problem right here? How is it going to glide while this repressor is located right here? Therefore, as long as this repressor protein is located and attached to the operator sequence, then what happens is those genes are never going to be transcribed and they're never going to be translated. What does that mean? It means that E. coli is not going to be able to metabolize lactose as long as these genes are not being transcribed and translated. And so to turn on the expression of these genes, what needs to happen is there has to be an inducer. Now, what is the inducer in this case? Remember that this is the lac operon, meaning that those are the genes that are going to help metabolize lactose. And so the inducer is actually going to be lactose itself. Either lactose or an isomer of lactose known as allolactose. What happens is when lactose is around, this lactose or an isomer of it, it's going to come and bind to the repressor protein. What happens is when it binds to the repressor protein, notice how the repressor protein changes shape. What's the big deal of the repressor protein changing shape? If it changes shape, then it's not going to fit within the operator anymore. And if it doesn't fit within the operator, then guess what? The operator is now free, and so the RNA polymerase is going to be able to bind to the promoter, and it's going to be able to glide freely along the DNA sequence, and nothing is going to be blocking it. So notice how, again, nothing is going to be attached right here to the operator. And therefore the RNA polymerase is going to be able to glide smoothly along the DNA sequence, and transcription is going to go ahead and take place. So these genes right here are going to be transcribed. You're going to have a messenger RNA, and then that messenger RNA is going to be translated, and we're going to get the proteins that are going to help E. coli absorb lactose and metabolize lactose. What are those proteins that are going to be produced? That is beta galactosidase, and that's the protein that's going to help the E. coli metabolize lactose. Then there's permease, which is a protein that's going to help E. coli actually absorb lactose. And then there is acetylase, and that's actually a protein that can also help E. coli actually break down other sugars other than lactose. So at the end of the day, for E. coli to really break down lactose and to metabolize lactose, those genes right here need to be, or those proteins need to be expressed. And so the only way to express those proteins to help E. coli metabolize lactose is lactose itself. Lactose is the inducer. Once lactose is around, then lactose is going to bind to that repressor protein, changing its shape, freeing up the operator sequence so that the RNA polymerase can glide easily and transcription can go ahead and take place. And that's why we call that operon an inducible operon. It's an inducible operon because it was originally, those genes were originally turned off, and only when lactose was around, or an isomer of lactose that's around, that is when this repressor protein is going to change shape and therefore freeing up that operator so that the RNA polymerase can go ahead and proceed with transcription, and then later on translation can go ahead and take place. And so that's why it's called an inducible operon, because you are turning it on. One thing to note is that if the bacteria is provided with two sugars, lactose and glucose, which one will the bacteria prefer to metabolize? The bacteria is going to metabolize glucose definitely faster and more compared to lactose. And that is because glucose is a monosaccharide. So the bacteria is going to have an easier time metabolizing a monosaccharide versus a disaccharide. Lactose is a disaccharide. So if the bacteria is provided with both sugars, the bacteria is going to prefer to metabolize glucose first. And so therefore, for the lac operon to be turned on and for the bacteria to go ahead and make the proteins needed to metabolize lactose, two things need to take place. One is lactose needs to be present, because once lactose is present, it will bind to the repressor, and therefore transcription of the genes that will encode for the proteins for lactose metabolism can go ahead and take place. But another thing is that the amounts of glucose need to be very, very low. Once the amounts of glucose is actually very low, a compound known as cyclic AMP increases in number. That compound right here, that molecule, cyclic AMP, binds to a protein known as the CAP protein, which stands for catabolite activator protein. And once it binds to it, it activates it, which then that active CAP protein binds to the promoter site of the lac operon, and that's going to help the RNA polymerase to bind to the promoter, and therefore the transcription of the genes that are going to encode for the proteins that would help the bacteria metabolize lactose can go ahead and proceed. If the bacteria is provided, as I said, with both lactose and glucose, and let's say the glucose the glucose amount that's provided to the bacteria is very high, then as I was saying, the bacteria is going to prefer to use the glucose versus the lactose. So as you can see, if the glucose levels are pretty high, and even though lactose is still present, and lactose binds to the repressor, and therefore it inhibits the repressor from binding to the operator, transcription of the genes right here that are needed to metabolize lactose, it's not going to take place. And the RNA polymerase is not going to bind to the promoter. What's preventing the RNA from binding to the promoter is actually the high levels of glucose. When glucose levels are pretty high, then cyclic AMP is going to actually be very low. And you can see right here, the CAP protein stays inactive. And without that CAP protein binding to the right here to that binding site within the promoter, then the RNA polymerase is not going to bind, and therefore the proteins needed to metabolize lactose are not going to be present. So as you can see, the lac operon is actually regulated by two things. One is the presence of lactose, and another thing is low levels of glucose. Because once the glucose levels are pretty low, then and lactose is present, both again are needed for the transcription and translation of the proteins needed to metabolize lactose can go ahead and proceed. And you can see right here, as I was saying, if you provide the bacteria with both sugars, glucose and lactose, you can see definitely that the bacteria prefer glucose and they grow rapidly when they are provided with glucose versus lactose, because it's just easier to metabolize a monosaccharide compared to other types of sugar. As I said, if the bacteria is provided with both sugars at the same time, the bacteria is going to prefer to use definitely glucose first. You can see that the bacteria is using glucose first. And then once the glucose is out and the bacteria has used all of it, then it's going to go ahead and move on to using and metabolizing lactose. But actually what you're going to notice is that there is a lag time after in between. So there's a lag time in between the glucose usage and the lactose usage. And the reason why there's a lag time right here is because as the glucose levels decrease, because the bacteria has used it, that's when the cyclic AMP levels are going to increase, activating the cap protein, binding to the promoter. So all of this takes a while before the transcription and translation of the genes responsible for encoding the proteins to metabolize lactose can go ahead and take place. So because there's that period of time where all of these events are taking place, you get that lag time. But once again the lactose binds to the repressor, and once the active cap protein binds to the promoter, then transcription and translation will go ahead and take place, and the proteins needed to metabolize lactose will go ahead and start using or allowing the bacteria to go ahead and metabolize and break down lactose. We have another type or another example of an operon, and this one is a repressible operon. A repressible operon, it's an operon that's usually turned on, so the genes are usually being expressed, and then they go ahead, these proteins get turned off and transcription gets blocked. An example of a repressible operon is the TRP operon or the TRP operon, which stands for tryptophan. So this is an operon that's responsible for it has genes that are responsible for encoding for a protein that's going to be needed in making the tryptophan amino acids. So as we said, an operon is going to have a promoter, the operator, and then those structural genes. And then we have the regulatory gene right here in front of the promoter. Regulatory genes are usually responsible for encoding for repressor proteins. So you can see, as I said right here, the trip operon is usually turned on. So usually transcription and translation of those structural proteins, it's usually taking place. And as I was saying, the trip operon encodes for a protein that's going to be needed for the tryptophan synthesis. Now, as I said, the regulatory protein usually encodes for a repressor protein, but usually those repressor proteins that are made, they are actually inactive, and therefore they are not blocking and they're not turning off this operon. And so therefore, continuous production of the proteins needed to make tryptophan continues to take place. And so what happens is as this operon is turned on and lots of proteins are being made to help make tryptophan, what ends up happening is the levels of tryptophan increases. And so the high amount of tryptophan actually ends up acting as a co-repressor. When the amounts of tryptophan increases, the tryptophan is actually going to bind to the inactive protein that's being, again, made by translating and transcribing and translating the regulatory genes. Once tryptophan binds to the repressor, and so we call tryptophan a co-repressor. Once it binds to the repressor, it activates it. Once it's activated, it's going to go ahead and bind to the operator, preventing RNA polymerase from moving forward, and therefore preventing those genes from getting transcribed and eventually translated. And what happens is that's going to go ahead and turn off basically that operon. So as you can see, that tryptophan operon is usually turned on, and then it gets turned off when the levels of tryptophan increases. When the level of tryptophan increases, it's going to act as a co-repressor. It binds to the repressor, activating it, which is then going to bind to the operator, and that's how you can turn off that tryptophan operon. So this is just a summary of an example of a repressible operon that's usually found in E. coli. So this is one way how E. coli can actually control the amounts of tryptophan that it produces. So as you can see, it does have an operon that helps the E. coli make tryptophan, but once the levels of tryptophan increases, the bacteria does not want then to keep that operon turned on. The bacteria would want to turn it off. So once the levels of tryptophan is really high, the tryptophan itself is going to act as the co-repressor, which is eventually going to turn off that tryptophan operon, and everything basically comes to a halt, and the production of tryptophan is going to stop. If you have a bacterial cell and you actually have parts of a DNA, so some DNA fragments that were obtained from a donor cell, from another bacteria, let's say, and you go ahead and you take that piece of DNA fragment and you go ahead and you mix it with some live bacteria, or dead bacteria, by the way. What's going to end up happening is actually this bacteria cell is going to intake that piece of DNA fragment, and it's actually going to recombination is going to take place where that piece of donor DNA is actually going to integrate itself with the recipient's DNA. And what's going to end up happening is now you have a bacteria cell that now contains genetic information that it didn't have before. So now this bacteria right here now carries extra genetic information. That extra genetic information could actually be very beneficial to the bacteria, because let's say those genes are antibiotic resistant gene, then you have a bacteria cell that wasn't resistant to antibiotics, and now it is, because of that recombination of genetic information that just took place. Griffith actually showed that this is possible. And the experiment that he did was he took some encapsulated bacteria, which means the bacteria that does have a capsule around it. And we've already talked about how capsules prevent phagocytosis. And so therefore, if a bacteria has a capsule, that increases its virulence, and it becomes more pathogenic if it does have the capsule. So what he did was he took some encapsulated Streptococcus pneumonia bacteria, and he went ahead and he injected those live encapsulated bacteria into the mouse. What ended up happening is the mouse did get infected, and eventually the mouse died. But they went ahead in the lab, they were able to take a sample from the dead mouse, and they were able to culture the colonies. And definitely those colonies where when they looked at them under the microscope, those bacteria were the encapsulated Streptococcus pneumonia. Then what they did was they got another mouse, and this time they injected the mouse with Streptococcus pneumonia, but this was a strain of Streptococcus pneumonia that did not have the capsule, so it was non-encapsulated. Non-encapsulated bacteria, they're not going to have the ability to evade phagocytosis as the encapsulated ones, and they're not going to cause disease. And so the mouse didn't die, and not a lot of colonies were obtained because most of them were killed by phagocytosis, but those colonies that they were able to culture on the plate, those were definitely non-encapsulated bacteria. Then the third step, what they did, they got another mouse, a third one, and this time they injected this healthy mouse right here with heat-killed encapsulated bacteria. So those were the bacteria that did have a capsule, but they basically heated them up, and the heat killed them. So when they injected the mouse with those heated encapsulated bacteria, the mouse did not get infected, and the mouse was healthy, and no bacteria were they were not able to isolate any colonies because those were already dead. They already injected this mouse with dead bacteria, so obviously, of course, no colonies were expected. Then what they did was they got a mouse, another one, and this time they injected the mouse with two bacteria. One is they injected the mouse with the live, these are live bacteria, and those are the ones that are non-encapsulated. So remember, the non-encapsulated did not kill the mouse, and they didn't cause the disease. So these were the non-encapsulated live ones. And then they went ahead and they also injected the mouse at the same time with the heat encapsulated killed bacteria. So those bacteria right here, even though they are killed, they still contain their DNA. They still have their DNA. And so what ended up happening is the mouse died. So that was definitely a surprise, because why did the mouse die? Even though this mouse was injected with the non-encapsulated. And you can see right here, when the mouse was injected with the non-encapsulated, the mouse was healthy, nothing was wrong. When the mouse was injected with the heat-killed encapsulated, again, the mouse was healthy. So why now all of a sudden the mouse died? What ended up happening is the DNA of the encapsulated heat-killed bacteria ended up actually merging with the bacteria in the healthy, or not the healthy, I shouldn't call them healthy. It was able to join in with the bacteria of the live, it was able to join in with the DNA of the live bacteria. Recombination took place, kind of like how I showed you with that previous slide. So recombination took place. The DNA within the dead bacteria joined in and integrated itself with the DNA in the live bacteria. And so what ended up happening is those non-encapsulated bacteria actually now they are encapsulated, and that's why the mouse died. They actually gained new genetic information, and that new genetic information allowed them to go ahead and produce and have that capsule around them, and that's why they were able to evade phagocytosis, and they were pretty virulent, causing the mouse to die. So it shows you right here, Griffith was actually able to show that DNA from a dead bacteria can go ahead and get integrated into another live bacteria, and that live bacteria is now going to obtain new genetic information that it didn't have before. This slide right here is talking about conjugation, which is a way of how bacteria can go ahead and transfer over some of their genetic information to one another. They project those sex pili right here that you see, and they can actually start transferring over their genetic information from one bacteria to the other. And again, this is really important because this is how bacteria can end up with new genetic information that they didn't have before. You can see right here an example of the conjugation taking place, for example, in E. coli, and how, let's say, this bacteria right here contains a plasmid, and usually plasmids contain lots of important genetic information, such as antibiotic-resistant genes are usually carried within plasmids. And you can see that this bacteria right here does contain the plasmid. This one does not, so it's like that's why they're referring to it as a negative cell versus this one as a positive cell, because it does have the plasmid. And you can see that it can actually pass over a copy of its plasmid. So it's not giving it up, it's actually just giving that other cell a copy of its plasmid. So now they both have a copy of the plasmid. An example of a plasmid is a plasmid known as the R factor or the resistant factor plasmid. And this plasmid, the reason why it's called resistant, because this plasmid carries lots of genes that are going to encode for proteins that are resistant to multiple antibiotics. So it contains tetracycline resistant genetic information, streptomycin. As you can see, like bunch of different resistant genes to so many antibiotics. And so if this plasmid gets basically transferred over to another protein that doesn't have it, then this new protein is now going to become resistant to these antibiotics as well. And so this is an example of a plasmid. Now, there are actually multiple types of plasmids, but this was just an example right here so that you guys could see how plasmids usually carry genetic information that increases the virulence and increases the pathogenicity of the bacteria that would carry it. We've already talked also about transduction previously, and transduction is another way of how bacteria can end up with new genetic information, and it involves a bacteriophage. So the bacteriophage, as it infects the bacteria cell, it goes ahead and injects its DNA, which gets integrated with the bacterial DNA. And the bacteria ends up with new genetic information that it didn't have before. Again, we talked about it in much more detail in the previous PowerPoint. So again, this is just a refresher of how, other than transferring over plasmids or conjugation, that's another way of how viruses can end up with new genetic information via transduction.